In the second part of this lecture on the material from the innate and nonspecific host defenses, we're going to start to focus some more on some of these things that some textbooks are going to classify over here in the second line of defense. So some of the different kinds of chemicals that you're going to find both on the exterior and the interior of animals. And we'll take a look at some of the effects of these chemicals, but also we'll see the cellular defenses that are built in and look at how there is an interaction between these chemical and cellular defenses that are out there that are, again, going to serve that role of the immune system in identifying foreign material that's out there and trying to remove it. And if we take a look in your blood here, first off, and look at some of the elements that we find in blood. This is just a cartoon showing what happens if you take a blood sample and spin it in a centrifuge. And you're going to get your separation into these three different distinct phases. You'll have your packed red blood cells down here. Probably about 45% of the total volume of your blood. And then you will have your plasma fluid up here on top that is described as sort of that color of hay or straw that's out there. And if you look in this plasma fluid, it's about 55% of the total volume, unless you have some sort of problem going on with your red blood cells, too many or too few. But within that plasma, it's mostly going to be water. And then you're going to find all these different kinds of elements that are being carried around in that plasma fluid, from proteins that are out there to clotting factors. You, of course, are taking nutrients out to your cells and likewise also returning the waste products. And you're going to have a variety of different kinds of things like gases that are being carried back and forth, like oxygen and CO2 in our example there. So there's a lot of elements that are being carried in this blood plasma here. And then we've got this last little fraction right here that's known as the buffy coat. And this is kind of the consistency of bacon grease that's solidified a little bit there. And this is where we're going to find the elements of our immune system that are involved in really finding and destroying a lot of material that's out there. And so we'll take a look at the white blood cells that you're going to find in this white layer right here. And if we look more specifically at some of these things that are floating around in the plasma fluid, and again, that idea on the chemicals that are found out there in that so-called second line of defense, once you get past the armor, we can find a variety of different kinds of acute phase proteins. So these are ones that are going to act really, really quickly. And you can see that some of these proteins will actually coat the bacteria that are out there, and that's really a signal for phagocytic cells to grab on and eat them. You can see we've got our blood clotting factors that are out there, like fibrinogen, that's going to be able to cause your blood to clot and limit the loss of your fluids there. And then we've got some more of these examples of these proteins that are floating around in your bloodstream, in this case, that are actually able to bind up with iron. And when we look at this process of iron, it's really kind of an interesting thing, because bacteria out there do have a natural transporter, this reductase protein that is going to basically bring that iron from the exterior of the cells into the cell so they can use it for their enzymes to function. And so if you think about this, the bacteria actually need this iron in order to divide. And when they get inside of your body, we are producing these iron binding molecules to try and limit the amount of available iron for microbes to be able to use. And so you can see we make ferritin protein. We've got hemoglobin in our red blood cells to bind it. You've got lactoferrin in blood and other bodily secretions. And we've got transferrin that's being produced. And so all throughout our body, we've got all of these proteins that are made to bind up to different forms of iron that are out there and prevent it from being readily available for the microbes. And so now you're a bacteria, you've got your reductase, but there's nothing in the surrounding area that's free that would allow you to transport it. So what do? Well, now you can make a variety of different things, like the hemolytic enzymes that we saw before in lab, where you break down the red blood cells and you release the hemoglobin, and that's going to allow the bacteria to then transport it. But another mechanism that they came up with that's really kind of intriguing that evolved is this idea of siderophore, which is a protein that will actually scavenge. It has a very, very high affinity for binding to iron that is floating around there. And so by making these siderophores, the bacteria are effectively able to suck the iron out from something like lactoferrin in your blood or in your tears or your bodily secretions. So you've got lactoferrin binding it, and along comes this bacteria factor that can actually bind it even tighter. So it is literally removing the iron from the places where we are trying to hide the iron from the bacteria. And so this idea of evolution with our immune system is really an intriguing kind of idea, that they evolve a mechanism, we evolve a counter mechanism, they evolve a counter to that, and it's going back and forth. But we're talking about millions of years of evolution going on here. We're not talking about something that is happening overnight. In addition to all of these different kinds of acute phase proteins, we can also find another set of antimicrobial peptides that are out there. They are relatively non-specific, and so we can consider them broad spectrum because they will work on a variety of different things. And if you look at this, you can see one overwhelming kind of theme when we look at these antimicrobial peptides is that they will disrupt the membrane. And what they are is basically short little, maybe 12 to 50 amino acid long peptides, or short little proteins that will have polar ends and then hydrophobic ends. So they will mimic your phospholipids that you're going to find in your membrane barrier. And these guys will basically start to poke holes in that. And so if you think about this, you have a bacteria cell out there, you poke holes, it's going to leak its cytoplasmic contents, and the cell is going to be more sensitive to osmotic pressure that's out there. But this is broad spectrum. It's going to disrupt a whole bunch of different membranes. For our cells, these things aren't going to affect it because we tend to recycle our membranes more effectively and quicker than you would find in some of these other pathogenic things that are out there. But really kind of an interesting thing to think about, that we're actually secreting these short little proteins into our intestinal tract, on your skin, or actually throughout your body so that it can get into your bloodstream, and you're effectively poking holes in things that are trying to get inside of you or have worked their way inside of you. So again, non-specific with broad spectrum kinds of antimicrobial peptides in your bodily fluids that are out there. Another example of proteins that we can find in your blood plasma is a group of proteins out there that are known as complement proteins. And these guys, you're probably talking about 30 different proteins that are typically floating around in your blood that are going to work collectively to actually destroy a bacteria or a viral infection. So it's really ubiquitous. You're going to find these proteins circulating throughout your body out there. And what happens is you can have 3 different ways of activating your complement that is out there. And we can see over here what's known as the classical pathway. When they first discovered these proteins, they found that an antibody could bind to a foreign particle or antigen, and that would actually trigger the process of what's known as the complement cascade. So once you activate, or once you have this antibody binding, that's going to trigger this complement protein that is then going to trigger this, and you've got a domino effect as you get all of these complement proteins starting to be activated by that initial activation event. There's a second pathway over here in what's known as the lectin pathway, and here you have a lectin that is actually able to bind to a sugar called mannose that we don't make, but it's a sugar that you can find on the surface of pathogens that are out there. So the antibody targets it, or something on the surface of your pathogen is actually going to trigger your complement cascade to start, and then you get your domino effect. And then they discovered an alternative pathway out there that is basically going to be a protein called C3 that is able to bind to certain structures on the surface of bacteria or fungi in the cell walls, or on the surface of different kinds of parasitic organisms. So a little bit different pathway, but ultimately all 3 of these methods are going to activate this domino effect. And when you look at this, well, okay, what happens as a result of this? Well, if we look at the classical pathway, you initiate by this interaction between this complement protein right here and this complex of your antibody bound to this foreign particle there. That's going to cause this complement cascade to start happening, and you're going to start amplifying the signal as this is going to interact with a whole bunch of these. It'll interact with even more of those, and you're going to get 3 different types of effects as a result of your complement cascade, your dominoes tipping over right here. And I found a little bit better picture to sort of show you how this process works. Again, don't memorize all these things. What I want you to think about is, how do we trigger this at the beginning right here, this activation step, either antibody or the sugars on the surface of these cells that are out there. And that's going to trigger your complement cascade where you amplify your signal. And if we look at the 3 results that we see when we look at the process right here, one of them is you're going to trigger these group of complement proteins that'll form what's known as the membrane attack complex, or MAC, and that's actually going to poke holes in the membrane of cells. So think about our antimicrobial peptides, same idea, but now we are directing this with proteins that are circulating throughout your bloodstream there, and that's going to cause your cells to lyse and die there. We're also going to have these 2 complement proteins are going to get released, and that is a trigger for the process of inflammation. So you're going to have a whole bunch of different things coming out of, not only are you destroying this particle, but you're also going to trigger inflammation, and we'll take a look at all the events that are going on there. And if you look at this part right here, coming off of this C3b protein that is able to coat things that are out there, you do what's called opsonization. And when you have something that is opsonized, this is like planting a flag on the surface of this particle saying this is a foreign particle, and then your phagocytic cells are going to be more likely to recognize that particle for destruction that are out there. So if we look at this, 3 different ways of activating complement, they all go through the C3 protein. So again, think about that idea of you can be immunocompromised because you have a mutation in complement proteins now. And here, if you have a mutation, and I had one student in the past who actually had a mutation in her C3 pathway, and she was more likely to get sick compared to people around her. Why? Because she was missing out on all 3 of these effects because she did not have she had an autoimmune disease where an antibody was being produced that actually bound to her C3 protein. So effectively, she was shutting off part of her innate defenses that are out there. If we look at another thing that's going on here, this process of sending signals throughout your body is going on during an infection. And we have cytokines that are out there that are actually proteins that are secreted by your cells, and they're going to have an effect on other cells that are out there. So really kind of interesting when we look at them. Some of them are known as interleukins, and we'll see this more when we look in the next chapter at the specific immune system. But you can see what interleukins do. They go in between white blood cells, and they're going to stimulate other parts of your immune system. We've got chemokines right here. Look at that chemical movement. This is a chemical that's going to be secreted, and that's going to cause white blood cells to actually move towards the site of these chemicals here. So they are doing this process, basically sniffing out these chemicals and then following them back to their origin. And then the last kind of cytokine or signal that we can see here is a group of proteins that were identified because they were found to be turned on after viral infections. And so they said, oh, these proteins must interfere with a viral infection, and they sort of do. And if we look at this process right here, you've got your cell that's been infected by a virus right there. And that virus is going to, in this case, release double-stranded RNA into the cytoplasm. And that'll get detected by this infected cell right there. And that is a trigger for that cell to actually start producing these interferons that are out there. And so we've got alpha and beta interferons and then gamma interferon. And those are going to be secreted into the surrounding area right there. And this is one of those chemicals that is actually going to attract phagocytic cells, or this is going to be released and bind to these surrounding cells in the area right there that actually do have receptors for the interferon proteins. And so this signal is getting released by the virus-infected cell that's going to die anyways, but it is effectively warning its neighboring cells, hey, you can think of it as my house is on fire, and it's polite to go and wake up your neighbors in the middle of the night and let them know so that their houses don't burn down. And that's what they're doing. They're releasing these interferon proteins right there. And if we look at the final effect right here, your alpha and beta interferon proteins, those are the signal that's going to turn on what are known as antiviral protein genes. And those antiviral protein genes are going to go in there and slow down translation in the cells and also start to target our double-stranded RNA that is in the cytoplasm for that cell. So that is kind of an interesting thing when we look at that's going to turn on some antiviral genes. This can also cause neighboring cells to basically commit suicide. So even if the virus does get in there, that cell is going to kill itself before the virus can replicate. That's alpha and beta interferons, and they isolated this third group over here called interferon gamma, and this is what actually is going to activate some of the surrounding immune system cells, especially your phagocytic cells. They're going to be on steroids, and they're going to start to clear it. But if you think about it, these cells have died, so now you've got all these dead cells floating around there. That is going to increase your phagocytic cells that'll go in there and clear out all of those dead cells and get rid of some of the cell debris from the viral infected cell. So that's a really good thing to have going on simultaneously. You turn on antiviral proteins and you turn on the cleanup crew to come in there. The problem with this is these antiviral proteins, when you do get them produced, they are going to cause side effects, what we see with flu-like symptoms. So the muscle ache that you feel with a viral infection like influenza, that's actually a result not of the virus, but actually your antiviral protein genes that are getting turned on are what are giving us that muscle ache associated with the flu virus. So we've been taking a look at a lot of these chemical defenses that we're going to find in the body here. And again, some people say it belongs in the first line. Some people say there is a separate second line of defense. It totally depends on your interpretation. But the important thing to remember is these kinds of physical and chemical defenses that we've been looking at so far, those are built in. They are relatively non-specific in terms of they aren't finding a specific species. Maybe they're looking for a family or a group of organisms that they will actually have their target. And in the next part, we're going to start to look at the cellular defenses that are built into this second line of defense.